Welcome. This is the continuation of the previous class, that is Androecium. We have discussed the structure of stamens, stamen cycli, etc. Today let us see the attachment of anther to the filament and the dehiscence of anther. Right? Attachment of filament to the anther. Based on this, we can see various type of anthers. Sometimes the filament is attached to the base of the anther lobe, then it is called basifixer. Sometimes the filament is attached to the entire length of the anther lobes, then it is called adnate anther. You can see in the case of Androecium, there you can see the adnate anther. And sometimes the filament is attached to the dorsal side of the anther lobes. Such anthers are called dorsifixed. Sometimes the filament is attached to the middle of the anther lobes so that the anther lobes can freely swing in the air. And this type of anthers you can see in the case of anemophilous flowers. Anemophilous flowers means the pollination takes place through the agent of wind. And such a type of attachment we call it as versatile anther. Versatile anther, so that a small breeze can freely swing the anther lobes and the pollen grains are liberated. And such type of anthers are called versatile anther. Sometimes the connective is divergent and anther lobes are attached far apart, and such type of anther is called divergent anther. Divergent anther. Sometimes the connective is long stalk-like body, and placed crosswise, right? Here you can see, and placed crosswise on the filament, separating the two anther lobes. And the longer one is fertile, and the shorter one is sterile. And such a type of attachment is called distractile anther. Distractile anther. And in addition, sometimes you can see some appendiculate structure just above the anther lobes. We call it as staminal corona, as in the case of Nerium. Staminal corona is also there. So these are all the various types of attachment of anther to the filament. Could you follow me? Then we have already discussed gynandrous type of anther. Gynandrous. That means gynandrous means the anthers are partially or fully attached to the carpels. Such a condition is called gynandrous condition. And sometimes, as in the case of Orchidaceae, orchids, anthers are fused to the stigmatic lobes. Anthers are fused to the stigma. And such a condition is called gynostegium. Gynostegium. The pollens are enclosed in pollinia there. Two mass pollen grains are there. And such a condition is called pollinia. And these pollinia are attached to an adhesive glandular disc on the stigmatic angle. There you can, in the case of orchids, there you can see this type of gynostegium and pollinia. Also you can see in Asclepiadaceae members, there also, here you can see the pollinia. So pollen sacs are there. Could you follow me? Then sometimes the Filaments and stamens are, no, the stamens, entire stamens are free from one another. Anthers and filaments are free. There is no fusion. And such a condition is called polyandrous condition. See, as in the case of lotus and rose, there you can see numerous stamens are there. And these stamens are free from one another. And that condition is called polyandrous condition. Could you follow me? Polyandrous condition. Then you can see the adelphous condition. What is adelphous? Fused condition of filament. The filaments are fused. Others remains free. There are three types of adelphous condition you can see. That is, the first one is monoadelphous condition. As in the case of Malvaceae, especially Hibiscus rosa sinensis. There you can see all the filaments of the stamens united to form a staminal tube, and here you can see anthers remain free, and such a condition is called monoadelphous condition, right? Monoadelphous condition. Then you can see the diadelphous condition. Diadelphous condition, as in the case of P, there you can see total 10 stamens, 9 stamens, the filaments fused to form one bundle, and one stamen remains free. And this condition, all the 10, total 10 stamens are there, 9 stamen, filaments fused to form one bundle, one stamen free. And this condition is called diadelphous condition. You can see in the case of P, right? And the third one is called polyadelphous condition. So in the case of citrus, there you can see the filaments of the stamens unite more than 2 bundles. Here you can see 5 bundles, and others remains free. Such a condition is called polyadelphous condition. Adelphous means the filaments fused in nature. That is what is called adelphous condition. Adhesion, we have already discussed adhesion is there. Epipetalous, epiphyllous, epitepalous, we have discussed in the last class. Here we can see the adelphous condition. Adelphous condition means fused condition. Fused condition of filaments, and others remains free. There are 3 types: monoadelphous, diadelphous, and polyadelphous condition. Right? Could you follow me? And sometimes, the anthers are fused, and filaments remains free. Anther heads are fused. So in the case of Tradax procumens, you can see this type of arrangement. That condition is called syngenaceous condition. The anthers are fused, see, and the filaments remains free. This condition is called syngenaceous condition. Then synandrous condition. Anthers and filaments fused together. Anthers as well as filaments united to form one group. Example, cucurbita, right? Then coming to dehiscence of anther. Dehiscence means splitting of mature plant structure. Sometimes fruit is also dehiscing. That is called fruit dehiscence. If anther is split open, then it is called anther dehiscence. Right? Dehiscence of anther. Splitting of a mature plant structure along a built-in line of weakness to release its inner contents. Right? Depending upon the position of dehiscing phase, we can name the 3 structures are there. Extros, that means the dehiscing phase facing towards outer side. That is called extros. Then intros, dehiscing phase facing towards inner side of the flower, center of the flower, that is called inroads. Then laterose, dehiscing phase facing laterally, that is called laterose, right? Depending upon the dehiscing phase, you can see extrodes, inroads, and laterose. Okay. Then there are four types of dehiscing, you can see. The first one is called longitudinal dehiscence. Longitudinal dehiscence means it's dehiscing along a structure parallel to the long axis of the theca. It is considered as the most common type in angiosperms, longitudinal dehiscence. Then poricidal dehiscence. Dehiscence is done by a pore at one end of the theca, maybe at the apex or at the base. If it is apex, then it is called apical pore dehiscence. If it is base of the theca, then it is called basal pore dehiscence. See, poricidal dehiscence. Then transverse dehiscence. The transverse dehiscence is exactly similar to longitudinal dehiscence. Only thing is, it splits at right angles to the long axis of the theca. It splits at right angles to the long axis of the theca. Then valvular dehiscence. So very interesting dehiscence in the case of Lauraceae members. The dehiscing through a pore covered by a flap. That is called valvular dehiscence, seen only in Lauraceae members. So we can easily identify the Lauraceae members by observing this valvular dehiscence. Right? These are the type of dehiscence you can see in the case of angiosperms. Could you follow me? See, you can see the type of dehiscence here. This is longitudinal dehiscence, and this is lateral dehiscence, laterose. The dehiscing phase facing towards outer side, this is called extrodes. Facing towards inner side, this is called inroads. Then transverse dehiscence at right angles to the long axis, that is called a transverse dehiscence. Then poricidal dehiscence, apical pore dehiscence, the pore seen at the apex of the theca. So this is called apical pore dehiscence. Sometimes you can see in the basal region, then it is called basal pore dehiscence. Apical pore dehiscence is there, basal pore dehiscence is there. Then it is called valvular dehiscence. Here you can see a flap like structure here, small opening, and it is covered by a flap. And this type of dehiscence is called valvular dehiscence. This is seen in Lauraceae members. Okay, will you follow me? With this, see, this is another, you can see the longitudinal dehiscence here. See, longitudinal dehiscence. And this is apical pore dehiscence. See, apical pore dehiscence. Right? Then this is Lauraceae member, Persea americana, Lauraceae member. Here you can see the valvular dehiscence, and small membrane is there, membrane covering the flap. See, this is valvular dehiscence, one of the characteristic feature of Lauraceae member. And with this, we can stop today's class. And the next class, we can discuss the innermost wall of a flower, gynoecium. Thank you very much.